Welcome to our webinar. My name is Linda Xue. I am Principal Scientist of Data Science and Bioinformatics at Crown Bioscience. The title of our webinar today is Preclinical Biomarker Discovery via in vivo and in vitro screening. Preclinical models, including in vitro biological platforms like cancer cell lines and organoid models, in vivo animal models such as mouse syngeneic models, mouse homographs, patient-derived xenografts (PDX) and cell line-derived xenografts (CDX, are widely used for oncology research and cancer therapy research before clinical trials. Biomarker analysis based on comprehensive omic profiling of preclinical models or samples collected from preclinical studies on different molecular levels can be a very important and cost-effective way to accelerate drug development and increase the overall success rate of the following clinical trials. To better select models for specific preclinical usage, we have a series of tumor model databases available, covering over 3,000 PDX models, 240 CDX models, over 1,000 cancer cell lines, and 500 organoid models, etc. Comprehensive data characterizing these preclinical models include pathology, pharmacological data, genomic and transcriptomic data, immunoprofiling, and proteomics data. Relying on these powerful data, we are able to perform preclinical biomarker discovery via in vitro and in vivo screening, which leads to the main topic of our webinar today. I will introduce large-scale preclinical screening by in vitro and in vivo models separately, and based on in vitro and in vivo outcomes, how to perform predictive biomarker research by using 4 case studies as example. First, since in vitro platforms are fast and cost-effective, they are always used to do large-scale screening of test drug efficacy. The biomarker discovery via in vitro screening is aimed to explore or validate drug mechanism of action, identify genetic features associated with drug response on a set of cell line or organoids, develop useful single gene or composite biomarkers, and eventually provide advice and guidance on future in vivo model selection. Streamlining with our well-validated large-scale cell screening platform, OmniScreen, we can quickly evaluate our test drug with large number of cancer cell lines and retrieve responders and non-responders from test cell line panel. Then via comprehensive bioinformatics analysis by combining efficacy data with genomic information of baseline cell lines, we are able to identify novel targets and hypotheses of the drug MOA, and also predictive biomarkers, which can be used for further in vitro and in vivo validation. Here is one example of predictive biomarker. BRAF mutation is used as a biomarker for MEK inhibitors that target MEK1 and MEK2 on the ERK MAPK signaling pathway. We can see that both two MEK inhibitors show lower AUC of dose response curves, that is, stronger cytotoxicity in the BRAF mutant cell lines than in BRAF competent cell lines, despite very clear AUC separation. Now I will talk about one case study based on large-scale in vitro screening of hundreds of cancer cell lines covering many cancer types. This study is focused on a chemotherapy drug, irinotecan. Irinotecan is a DNA topoisomerase 1 inhibitor that interrupts the cell cycle in the S phase, so stops DNA replication initiation and causes cell death. SN38 is the active metabolite of irinotecan. The aims of this study is first to assess drug efficacy across various indications using cell lines. Second, to investigate drug MOA and validate the MOA on a historical mouse clinical trial data set, and also develop precision composite biomarkers for further preclinical usage. Here we plot the distribution of 300 cell line response represented by AUC values separated by tissues of origin. We observe significant difference across cancer types, and irinotecan is more effective in blood cancers than solid tumors. If we remove blood cancer cell lines, the variance of in vitro response in all these 257 solid tumor cell lines are relatively small. In this case, we would perform separate analysis for blood cancer and solid tumors. Blood cancers are, of course, with our interest because of better response, but here we will only show you the results on a much larger cohort of solid tumor cell lines for more statistically powerful analysis of biomarker discovery. The solid tumor cell lines were then classified by proper AUC cutoffs according to overall distribution of the cell line sensitivity to the drug. As we can see, 86 cell lines with lower AUC were defined as sensitive cell lines, while another 86 cell lines with higher AUC were defined as insensitive cell lines, and 85 cell lines in between were grouped as median cell lines because we are not sure these are true responders or not. Transcriptomic analysis on these solid tumor cell lines indicate that there is no obvious clustering on sensitive, medium, and insensitive groups that we defined from the in vitro outcome based on expression of all 20,000 human genes. So specific molecular biomarkers need to be found to separate the compound efficacy. By comparing sensitive and insensitive cell lines, we identified the top number of differentially expressed genes And no matter how many genes we select from top 100, 200, 300 to 1000, these genes are always enriched in DNA replication and with the most significant p-value, which directly point to the mechanism of the drug. We also confirm this result with pathway analysis across 200 canonical pathways that cell cycle related pathways like hallmark G2M checkpoint pathway are significantly more activated in sensitive cell lines versus insensitive cell lines. Next step, we collected our own historical in vivo dataset for irinotecan. This is a two-arm mouse clinical trial of 16 PDX models involving colorectal cancer, gastric cancer, and lung cancer, 3 solid tumor types. By linear mixed model approach applied on in vivo dataset, we could verify the in vitro findings that the most significant goal term and the pathway for the top genes also indicate the MOA of the drug. So the most significant goal term is DNA replication initiation, and the pathway is cell cycle. Back to the in vitro dataset, we identified SLFN11 as the most significant single gene expression biomarker that are highly upregulated in sensitive cell lines. SLFN11 irreversibly blocks replication in cells under replication stress. And its high expression is common single gene biomarker to DNA targeting drugs, which has been reported by different literature. However, we have more sophisticated method to investigate composite biomarkers that work more precisely in prediction of accuracy. This approach requires a large number of samples to divide the complete cohort into a discovery set and validation set, and then identify features best dissecting sensitive and insensitive groups by hundreds of permutations. The best combination of genes will be selected and validated in the independent validation set. This plot shows how we select the best number of genes. We test through from 5 genes to 30 genes, and we can see that a set of 21 genes shows the optimal prediction accuracy. By using these 21 genes, we can clearly distinguish sensitive and insensitive cell lines with 89% of prediction accuracy. 14 of the 21 genes are upregulated in sensitive cell lines, and SLFN11 is also in it. And 7 genes are in the other way around, so they are upregulated in insensitive cell lines. Not only in discovery set, The 21 gene panel also performed well in the independent validation set with 79% prediction accuracy. If we also include the median group cell lines that were excluded at the beginning of this analysis, we still can end up with high significant correlation between the 21 gene signature score and the AUC values of all 257 solid tumor cell lines. Then, if we compare the composite biomarker panel with the single gene biomarker, that is SLFN11 expression, it is very clear that the composite biomarker shows much better correlation with AUC because many cell lines are not expressing SLFN11 at all. By using multiple genes, we can strengthen the predictive power, especially in large data sets. With the first case study, irinotecan efficacy was assessed across cell lines and solid tumors were selected as a cohort for mechanism probing and biomarker discovery. Mechanism investigation indicates DNA replication is associated with efficacy in vitro dataset, and in the meanwhile, the validated in vivo dataset. We found that expression of SLFN11 is the strongest single gene biomarker, and also an expression-based 21 gene composite biomarker achieves higher efficacy prediction accuracy. The next case study I will talk about is based on smaller sample size cell screening, focusing on specific indications. So with relatively small number of cell lines tested, we are also able to identify useful biomarkers. This study is a collaboration project between CrownBio and Datum Therapeutics, and the results are published in Anti-Cancer Research this year. We were working on a fourth-generation platinum compound, cetraplantin. Cetraplantin is with a more favorable toxicity profile, especially increased in hematopoietic cancers when compared to cisplantin. In this study, we assessed cetraplantin and cisplantin efficacy across various indications using 66 cell lines, and most importantly, performed a systematic biomarker analysis to identify hematological malignancies with better response to cetraplantin. The AUC distribution revealed that the hematopoietic and lymphoid cancers show much higher sensitivity to cetraplantin than the other test solid tumors. Next, by gene expression and mutational analysis, we identified MTAP underexpression and BCL2 mutation are associated with cetraplantin cytotoxicity to hematopoietic cancers. On a DNA copy number level, we also observed that deletion of MTAP CDK and 2A, CDK and 2B, 3 genes located at the same genomic area, 9p21, together with DMRTA1, is associated with cell line sensitivity to cetraplantin, which agree with the results on the gene expression level. On the right panel, the IC50 values of hematopoietic and lymphoid cell lines are shown, and the cell lines with MTAP deletion are highlighted in orange and tend to be with lower IC50. In the second case study, we confirmed that cetraplantin is significantly more active to blood cancers other than solid tumors comparing to cisplantin. MTAP underexpression and BCL2 mutation are associated with cetraplantin cytotoxicity to hematopoietic cancers, and 9p21 copy number deletions, including MTAP deficiency, were identified as the key statistics, the key characteristics for high sensitivity to cetraplantin. Okay, how about in vivo case, which is more close to clinical phases? In vivo screening using mouse tumor models or human tumor xenografts can mimic phase 2 clinical trials to provide accurate measurement of drug efficacy and is powerful for biomarker discovery due to its unique study designs. Biomarker discovery via in vivo screening aim to explore or validate the drug mechanism, identify genetic features associated with the drug response on a set of in vivo models, and develop single gene or composite biomarkers, and also provide advice and guidance on future clinical trials. Our methodology has been published in BMC Cancer in 2019 as a cover story. This is a schematic design for a typical mouse clinical trial. A variety of patient tumors were collected and transplanted into immune-deficient mice. Multiple PDX represent intertumor heterogeneity, while the biological replicates within one arm introduce intratumor heterogeneity of original patient tumors. The design of mouse clinical trial allows for better efficacy measurement and biomarker discovery. Another widely used preclinical in vivo screening platforms is MuScreen, which is more efficient for evaluating immunotherapies because these are immune-competent mice with complete tumor microenvironment. So in vivo studies, the methods for biomarker discovery are a little bit different comparing to in vitro cases, because the experimental results are different. So I will give you an example to introduce in vivo biomarker discovery. This case study is about EGFR expression as a well-known single gene biomarker for cetuximab in gastric cancer. In a gastric cancer mouse clinical trial, we observed that EGFR overexpression and amplification are highly associated with a good response to cetuximab, and we confirmed this results by DNA level, mRNA level, and also protein level. To identify biomarkers, we used different methods, which gave us a clearer conclusion. First, tumor growth inhibition, TGI, as conventional in vivo efficacy measure, showed a weak positive correlation with EGFR expression, and the EGFR ranked as the 157th place within 20,000 genes in the human transcriptome. AUC ratio is the ratio between AUC of tumor growth curve from treatment group and AUC of tumor growth curve from vehicle group. Lower AUC ratio means better efficacy. Based on AUC ratio, we could see a better correlation with EGFR expression, and the gene rank as at the second place. However, if we use a more sophisticated statistical model, that is linear mixed model, to evaluate the association between gene expression and tumor growth, we identified EGFR as the top gene, which is much more significant gene than any other genes in the transcriptome. So we are able to verify EGFR expression as the top gene expression biomarker for cetuximab in gastric cancer. No matter the biomarkers were found based on in vitro or in vivo screening, we are mostly interested in how can we translate and apply them in clinical trials. So this is one example that we identified hypothesis-free biomarkers based on mouse clinical trial of 77 PDX models from 4 cancer types for a certain drug, which is ready for phase 2 clinical trial. We propose 6 biomarker candidate sets on mutation and gene expression levels. For instance, BCS class 2 is a mutational biomarker set used as current enrollment criteria for phase 2 clinical trial of the drug, which is also identified by us. And BCS class 4 is their newly added enrollment criteria based on our findings. But BCS class 1, which is a mutational biomarker set with multiple genes and better separates responders and non-responders with higher resolution, was not selected. It might be due to the fact that single gene biomarker can be more easily tested in practice than composite biomarkers. So the balance between predictive power and the operational convenience is very important for valuable biomarkers. To sum up preclinical biomarker discovery, a sufficient number of preclinical models with spread in efficacy is required for biomarker discovery. Comprehensive biomarker analysis is needed in biomarker discovery as well as drug MOA exploration and validation. Ideal biomarkers are numerically predictive, biologically interpretable, and operationally convenient. Single gene biomarkers are ideal but hard to find in practice, while composite biomarkers can be robust options. The potential biomarkers developed can provide insights and guidance on future in vivo model selection or future clinical trials. That's all my presentation today. Thank you for your attention, and I'm happy to take any questions.